peptides that are being formed from the P to the A site at the ribosomes, it's important to know what a peptide bond will look like. So you have at every amino acid, just like in every protein, you have an amino site and you have a carboxyl site. So on one side here you have a free amino group, and then the other side you have the carboxyl group. When you make the peptide bond, you are joining the carboxyl to the amino group. You are actually running a dehydration reaction, and you are getting this N-carboxyl type of bond.
C is for clue, that's okay with me.
Yes, this is very important to know. When you are elongating the peptide chain, you're making the peptide bonds, you have the amino acids that are formed, right? The peptide bonds that are formed, and they are catalyzed by something. What is it that is catalyzing this formation? This is important to know. It is the ribosomal RNA. It is in the large subunit of the ribosomal RNA. And remember that I had mentioned before that the problem with the chicken and the egg, which one came first, DNA or protein? And then remember I had mentioned about the central dogma, that you have the transfer of information that goes from DNA to RNA to protein. Now, a little bit more about the central dogma is that it is a one-way road. It does not go backwards. The information goes from DNA to RNA to protein, but it does not go back from protein to RNA to DNA. I mean, you can look at a protein sequence and you can now in vitro make your own artificial synthesized messenger RNA, but naturally it's not going to happen. Okay, actually, did I mention that we are the only species that know our own recipe? Right? Think about it. We are, after the genome, the human genome, we are the only species that know the recipe for making humans. I mean, not only in the practical term, So, I mean, in the intellectual point of view, DNA-based. So here you have, when you have that central dogma, and you have the question of which one came first, the protein or the DNA, you know that at certain point you had a type of an RNA world, because ribosomal RNA or some RNAs have catalytic activity. They are acting as if they were proteins. They are actually pushing a reaction to happen. This is catalytic activity. So the amino acid formation, the peptide bond formation, is catalyzed by a ribosomal RNA. It's not another protein, it's RNA. Now, looking at the termination, how you come up with to the end of this protein chain, you have the termination codons, UAA, UAG, and UGA. So also to finish up the chain, you have to have termination codons, and you also need release factors. So here, as I mentioned, for the elongation,
C is for clue, that's okay with me.
you have two amino acids, and you have a dehydration reaction, and then you have this peptide bond formation. Okay, so that's part of the elongation. Okay, and you have all your peptide chain that comes out of there. So now looking at the termination, I found here one codon that does not code for anything, that it says stop. At the end of your messenger RNA, actually it's not quite at the very end, but way before the poly-A tail, you have some type of a stop codon there, and it's close to the 3' end. If you forget, you just remember poly-A is at the 3', because the cap is at the 5' cap, and that's at the other side. Okay, so 3' end of messenger RNA. You find this stop codon, and how is it you're going to take this thing apart? So stop codon right there at the acceptance site, and it does not bind to another tRNA, it binds to release factors. So now these release factors will come into the arrival site here and to the A site, and what they're going to do, they're just going to dissociate the whole thing. They help with the dissociation of the polypeptide from this tRNA at the P site, see? Once these release factors come in, polypeptide is broken free, and then you have here, polypeptide is out, and then you have the dissociation. And also, this thing was kept together. It needs some type of energy to break it apart. So in order to dissociate here, you also use energy in form of GTP. So you have, again, GTP hydrolysis. And you dissociate the whole thing, and everybody is ready for another round of protein synthesis. So here for the termination, in the written word, we have no tRNA that is corresponding to those anticodons, and what you have are the release factors. They bind over there, dissociate peptide, dissociate the other tRNA, and dissociate the whole complex. Here, you know you are missing GTP, but you know that this needs energy. This needs a GTP energy. Okay, just another little review, just exactly what we were talking about. Now this is just a little chart when you are studying and you want to have everything in one place and you want to find what is the process that comes first, what is the most important steps. So I just put that little chart for you. Now in this process here, which system is this, prokaryote or eukaryote? Okay, I'll give you a clue.
C is for clue, that's okay with me. This is in the bacteria.
You have transcription right there. What is transcription? Right, from DNA to RNA. Now, if you have transcription side by side with translation, it's telling you that there is no nucleus. So this is in a prokaryote system. Now, you see here, when you have a coupled transcription and translation, you actually can have this image here that you have here, for example, the gene, right? And you have close to this RNA several peptide chains that are growing. And these ribosomal RNAs, they got together at the 5' end, they start to make the peptide and they go towards the site of transcription. So you see, as you get closer to transcription, your polypeptide gets bigger. They get bigger and bigger and bigger. And also, you don't really need to have only one system of ribosomal RNAs here. You can have several of them, and they just go, one after another, making this polypeptide chain. So if you just imagine, a lot of people, they are doing crochet. Okay, remember old style crochet type? Anybody did crochet in their lives? Yes? Yes, I used to. One time when I had some time to think. So I actually enjoy making the patterns. Okay, so making crochet here, I start in the beginning, crochet is my polypeptide chain, and I am reading my DNA that is telling me exactly what to do in these patterns. So I move here, and it so happens that somebody else likes to do crochet too, so they hop on my little sequence of DNA that is telling me what crochet to make, and they write on my side. Now if I'm making my crochet here and I'm moving this way, my line is getting bigger, and my line is getting bigger. But this person just started. So they started right at the beginning. The line is still small. The crochet is still small there. By the time I get to the end, my whole crochet is done. But the ones that are starting after me, they have shorter and shorter crochet lines.